Thanks for the invite. So my name's Aaron, and I work at Trent University, and so this is about 2 hours northeast of Toronto, just to get you situated. And all of this work takes place on the traditional treaty and territory of the Mississauga and Anishinaabe. And what I want to talk about today is this question that I've been wrestling with for a while now, is trying to find meaningful genomic metrics, specifically with respect to small populations and populations of conservation concern, taking into account demographic history. And so I thought it'd be worthwhile starting with what I think the problem is. And I think there has been a lack of differentiating genome scale data from genetic data when it comes to small populations and conservation issues. And what do I mean by that? So what I mean by that, and I'm an editor at Conservation Genetics, what I see now are genomic papers that are just applying microsatellite analyses, structure, PCAs, and saying, hey, this is a population of concern. And I think that's a problem because we're missing an opportunity to take advantage of novel metrics and the power of genomic data. This problem also exists because with genome data, like you've seen today, there's a lot of new metrics: genetic load, runs of homozygosity. Those of us, Bridget and I, microsatellites, if you did microsatellites, I just see you. But, everyone sort of accepted what good values were and what bad values were for conservation. And that's not the same with genomic data. We're still wrestling with that. And moreover, some of these new tools, like depending on the way you estimate genetic load, if you do something like GERP, this is insanely computationally intensive. You're looking at 20, 30 different reference genomes on top of whatever you've sequenced. So it's quite challenging. In addition, we know when we estimate these summary statistics, we really need to account for demographic history. The magnitude of that bottleneck versus how long a population spent in that bottleneck is going to influence diversity. You can crash and recover quickly and we'll never detect it. Has purging happened? This is the new background selection. It's the norm, it seems. Given enough time, purging may have happened. And I think also this problem has seen renewed interest largely, I think, more recently due to the meeting in Montreal, the CBD meeting, where we're seeing a lot of people talking about this. And this is from Chloe Schmidt. I think it summarizes it nicely. Essentially, in her paper, she showed, hey, genetic diversity doesn't correlate with IUCN Red status. So what does this mean? We need to develop and assess metrics specifically designed to assess genetic diversity and inform conservation policy. We're still struggling with that. And so I thought where we could start with summary statistics is looking at effective population size. And Krisha, where's Krisha, did a great job summarizing this, and maybe I'll get the terminology wrong, but NE to NC in particular, NE, I agree with Wapel's most important statistic we can generate, diversity. This metric, specifically relative to census size, has a long history in conservation. The contemporary NE to NC, we've been looking at this for 20, 30 years. More recently, we've seen that this long-term NE or coalescent NE also may have a link to conservation status. Now, this contemporary metric gets used a lot, but it's highly controversial. Sean Hoban summarizes it nicely. Variation is large, in part due to what Krisha showed. Depending on the NE method you use, you're going to get a massive difference in value. That being said, without a better metric, let's still stick with this ratio of 0.1 being problematic. If you're below that value, that's a red flag, until we get new genetic metrics, or account for demographic assessments, highlighting the problem. We still got to figure out new metrics accounting for demography. More recently, and I was a part of the group on the left here, we've seen a link to with historical NE to NC ratio and conservation status. And so what we did on the left here in pinnipeds is we built a model looking at ancestral NE to NC against Tajima's D, and we made it more complex than we needed to do, but essentially the residuals, so if you deviated from that model as a pinniped, if you had a higher residual, meaning your NE to NC ratio was higher than the model predicted, you tended to be a species of conservation concern. High NE to NC values are problematic, and that was just shown by Wilder et al. Again, the dirty secret here, and I'm a part of this, so I'm okay to share it, is this is driven exclusively by NC. Nothing to do with genetic data. And that's what IUCN models or rankings are often based off of, is NC. And so taking that a little bit further, we were working at caribou, and what I want to highlight is that ancestral NE to NC value that is linked to conservation status. In this case, high values are bad. Really nice correlation with Tajima's D. Gaspésie, you may have seen this on the news. This population will be extinct in your lifetime. High Tajima's D, high NE to NC ratio. Important thing here, because maybe, hey, Tajima's D could be that summary statistic that we should be using to identify populations of concern. More recently, we've seen a lot of this today. With the advent of genomic data, we started looking at genetic load. Maybe this is also another metric we can integrate into our assessments. And I love this paper by Van der Valk et al. Out of Uppsala, Stockholm? Yeah. But what they showed and what we've seen today is across mammals, some massive taxa, those species with high diversity have the highest genetic load. Those species with low diversity have the lowest relative genetic load. So over long evolutionary time spans, if you're lucky enough to survive as a species, chances are purging has happened. Van der Valk also very nicely highlights a potential problem. So if we're going to use, or if we're going to consider genetic rescues, maybe we should be considering genetic load when we're selecting animals. And maybe we don't want to introduce the most diverse animals, or we need to at least consider that, because they're going to have a lot of deleterious alleles. Marty Cardos, we've seen this slide again as well. Doing this on orcas, similar pattern, a bit more of a mixed bag. We see an increase in ROH, and increased homozygosity mutational load. But it's not consistent across populations. Demography is playing a role. Some populations appear to be purging more than others. And he highlights that here nicely. Probably due to the stronger genetic drift associated with the smaller long-term history of NE. So we need to interpret things like ROH, genetic load, in light of long-term effective population size. And so my group's been interested in this, and so we looked at 3 different ungulates in North America: caribou, mountain goat, white-tailed deer. We'll just focus on mountain goat and white-tailed deer. Mountain goat, NE of about 100, and it's been this way for 30,000 years. 25% of their genome runs of homozygosity. White-tailed deer, NE tens of thousands. When we look at mutational load, mountain goat, basically nothing. Deer, double the genetic load of a mountain goat. Explanation, purging. Mountain goats have been like this for 30,000 years. They've had time to get rid of those alleles. We look at things like DNDS, again, extremely low value with mountain goat, consistent with purging not happening in white-tailed deer. And so I think collectively, when we're thinking about the genomic health of small populations and that problem, we really need to consider all of these metrics in light of temporal NE and NC, demographic history. And I think collectively, things like ROH, mutational load, and Tajima's D can give us insight on what's happening in that population and give us information. And I just want to remind folk, and I hate to be a naysayer, but when we think about these metrics, I've always been inspired by Joel Berger, if you know of him, a very influential conservation biologist. They wrote this paper about climate change projections. And the argument here is it doesn't matter unless we deal with contemporary problems. Exploitation, land use change. We can model as much as we want, but that's not gonna, if the species aren't there, it doesn't matter. And so I think about that a little bit with genomic metrics, that if we have the choice of buying land or sequencing a genome, we probably should buy land. So just keep that in mind when we think about management of small populations. So I want to show you some new work done by a PhD student in undergrad in my lab looking at deer, and I'm pretty stoked to present this. We've never shown it to anyone. Deer are odd to present this meeting because there's 20 million of them. But I hope to convince you that there's value. And we're going to talk about small populations and what we're seeing. And so today I'm going to talk about the white-tailed deer found pretty much in eastern North America. They do range out west, but there you start to see more and more mule deer. We've done about 90 whole genomes of the 2 species, but we're just going to talk about white-tailed deer, Odocoileus. Very interesting animal to work with because humans have a very long history with white-tailed deer. The first peoples in North America, especially in eastern North America, deer were a primary protein source. And so various evidence at times would suggest 70% of various indigenous groups, their diet was consisting of deer. When Champlain entered Canada, and so this is in southern Ontario, where close to where I live, the Neutrals, which were an Iroquois tribe, he came back with this drawing and suggested deer farming was happening. Deer farming wasn't likely happening, but it's clear that deer were critical to the local community. And we know for various reasons, deer have been historically used for food, shelter, clothing, and are of cultural significance. Now, when Europeans colonized North America, the relationship with deer was ratcheted up a notch. And what's cool about deer is it is the first example of a wildlife management regulation. So in 1646 in Rhode Island, because of overexploitation, they institute hunting seasons. This didn't exist. Over the course of the next few hundred years, deer were heavily harvested. From 1755 to 1773, 600,000 white-tailed deers were shipped out of this port to Europe. And this continued up until about 150 years ago, and I think most people in North America don't know this, deer were virtually wiped out. They were extirpated in most states, or what was left was fewer than 100 animals. Human history, modern day, is also very interesting with deer. They impact property values. So in Kansas, they're highlighting deer on this property. Anyone from Wisconsin? No? Okay, Wisconsin. There are houses whose property value is driven by deer. So this is a $1 billion industry in North America, hunting and all the residual effects from that. In addition, deer are an insane reservoir for COVID. There are places in the US where 40% of deer test positive for COVID. I've handled about 200 deer in the past 3 years. Thankfully, only 8 have come back seropositive. But they're a big reservoir. And in fact, in Ontario, we had COVID jump from deer to human. And so this is of massive interest when it comes to our health. Now, in terms of white-tailed deer and their long-term effective population size, and let's just remind you what Matt Hahn said, ignore the numbers, big trends are what matters. What we see with white-tailed deer, and we're just going to focus on this purple line, over the course of 2 million years, they've done pretty well. A massive crash that coincides nicely with the last glacial maximum. We see this a lot in virtually every PSMC plot. They come out of the last glacial maximum doing quite well in terms of effective population size. When we look at the more recent NE, what we see is white-tailed deer doing very well, and then about 150 years ago, their effective population size bottoms out. What's wild about this and why we're so excited is every single dot here corresponds to the date, the recorded date of a population low in any given state. So Alabama, the population low was about 120 years ago. The gray bars correspond to the stalking efforts. So we caused this collapse in census size, panicked, as we should have, and then we started restocking for 30, 40 years. So that's when the stalking started, that's when the stalking ended. So our new estimates, our new temporal methods of NE, like the fin whale presentation, we're picking up remarkably a human-induced collapse corresponds perfectly with the census data, and then a recovery that corresponds perfectly with our census, with our stalking efforts. Pretty awesome to see. Why does this matter? I want to bring you back to the Van der Valk warning, and if hopefully Andy Foote watches this because I credit him with this idea. We were interested in the effect this stalking had on genetic load and runs of homozygosity. And it turns out, if we look at inbreeding on the x-axis or genetic load here on the x-axis, there's a positive relationship between the last time we stalked that population, the last time we went and messed around with that population, in inbreeding and genetic load. So what Van der Valk warned about seems to have happened in white-tailed deer. The trend's noisy, I'll give you that, but it's scary, right? If our stalking efforts, if our meddling actually increased inbreeding and genetic load, that's something we need to consider. And so just to summarize this first part, is these temporal estimates of NE are amazing now. The power we have, and in our case, they coincide perfectly with exploitation, and deforestation was a big issue too, and stocking efforts. And I think important for this meeting, when that population was at its low, hundreds of animals to tens of animals, and we started restocking it, that has an effect on mutational load and inbreeding. And this is occurring in a population with a census size over 20 million currently. And so maybe in a small population purging might happen, but I don't know. But it's a worrying signal because we're seeing that effect. We've continued the white-tailed deer work focused on two small populations. So the key deer off the coast of Florida with collaborators in Florida. This is an endangered species federally. And then St. Pierre and Miquelon. So who's seen Peaky Blinders? That's the only reason you may have heard of this population. But St. Pierre and Miquelon, it's a French colony or French island off the coast of Newfoundland. Deer were introduced here in the 1950s. Deer became isolated here probably 8,000 years ago. They're interesting for a lot of reasons, and I won't talk about that today, but phenotypically, the key deer, about half the size of the mainland, so there's something happening selective-wise with the island. St. Pierre and Miquelon deer, high frequency of malocclusions, so jaw underbite or overbite, and leucism. About 10% of the animals manifest one of these two traits. And we know in deer that there is a fitness cost to this. Makes sense, right? This animal, if you're a wolf, you see that animal and forage. And so there's interest from local managers and more generally about the population genomic health of these two populations. When we look at their temporal NE, what's really interesting is key deer, they've been low for a long time. This is important because around the 1950s, there was a bit of worry about overharvest. We don't pick that up at all. We see this continued in the PSMC plot. It's been low, it's been constant for hundreds, if not thousands of years. St. Pierre and Miquelon, on the other hand, what we pick up is the introduction happened about 1953, so 80 years ago. And because our genomes carry that historical signal, we pick up the source population that was highly diverse, the crash on the mainland, then we pluck the animals and move them to the island, and their demographic, their NE, never recovered. And so we don't see, in contrast to the mainland deer, we don't see that recovery. So key thing to note here, SPM, St. Pierre and Miquelon deer crashed, never recovered. Key deer have been that way for a while. When we look at runs of homozygosity, we see elevated runs of homozygosity in deer in St. Pierre and Miquelon. Our control animals, we have an island to compare to, and what we've divided north and south mainland white-tailed deer. Again, this is around 90 individuals. We see elevated rates of runs of homozygosity. When we look at metrics of genetic load like missense and loss of function mutation, we again see elevated numbers. What's interesting about this is we know deer have a high genetic load. If you've seen the recent Oryx paper in Proceedings, I was particularly interested in their numbers, and it's dangerous to do this comparison, but our absolute values of missense mutations are considerably higher than what you see in the Oryx in the key deer. So slightly elevated mutational load, nothing on the mainland, and slightly elevated inbreeding. When we look at Tajima's D, what we see is St. Pierre and Miquelon have a high value relative to the Florida Keys. So they, Tajima's D, a positive value, loss of rare alleles. Florida Key, positive, so they've also lost rare alleles. Negative in our comparators. This is due to the magnitude of that change. We sort of had the double whammy with St. Pierre and Miquelon. The mainland crash, followed by the introduction. Florida Keys, likely 8,000 years ago, you had that bottleneck. Mean runs of homozygosity, slightly elevated. But what's interesting about the runs of homozygosity is we have elevated values in Florida Keys, some 10% of the genome, but they're not any longer. And we've heard about this today, and you can age these. These all occurred, the co-ancestry, we're looking at 200, 500 generations ago for those runs of homozygosity. So there's more runs of homozygosity in these, quote unquote, inbred populations, but they're not any longer. So that's due to presumably a founder effect, drift, not contemporary inbreeding, an important finding. And so collectively, when we bring this all together, we see that Tajima's D is reflective of the magnitude of demographic change. And in both those island populations, if you believe what we showed in pinnipeds in the Wilder paper, that Tajima's D value is worrisome. Higher FRH does not mean longer FRH. So we're not picking up inbreeding. It's a good thing. There's elevated mutational load in both of these populations with no evidence of purging, or clear evidence of purging. We have to dig a bit more into that. And so getting back to that initial question I had about the problem, do these metrics collectively suggest these populations are at risk? Are these metrics collectively a problem? And a bit of a lackluster ending, I don't know the answer. I don't know what I would tell managers about these populations. And I think, and I've always been inspired by Marty Cardos and his persistence on trying to link genomic metrics to things like population viability. And that's really what I think we need to do. I can show you that these populations have worrisome genetic signatures, but much like the tigers, we need to eventually link this to phenotype or population viability, I think, to convince people on the ground. With that, I will leave you. This is a deer that's sleeping, so that's the deer snoring. But you can watch us release a deer that we captured. I'll put it on mute, and I think I have time for questions. Nope.
Oh, yeah, if you do that, there's a fall.
Yeah.
There we go.
So we're just reversing it. And so she's about to wake up, and you'll see her wake up groggy, but I think I can take questions.
You popping up. There's a couple back here. One up here.
No, start here.
Oh, okay. There. You have to unmute here.
Okay, so
we want to mute it.
We want
for
the video.
I think to hear this, it may.
Oh, to hear that, we'll have to keep it on. Can we go back one slide, maybe, or?
Yeah, or we can just go.
There we go. Perfect.
That was the bonus slide. I can talk about this too. Yeah.
yeah, I was curious about, so with your inbreeding estimates, you said that like most of the runs of homozygosity are really short, and the FROH for those really small populations is actually surprisingly low. Do deer, is it known if like deer have some like behavioral response where they like try to avoid inbreeding? Like I think this was seen in hyenas or some other animal. I don't remember off the top of my head, but.
So I think the life history, deer do very well. And so I don't know of active inbreeding avoidance. No one's ever really documented that. But what I can say is, most yearling does are going to reproduce, and then most adult does, if conditions are well, are going to have twins. And so numerically, and you can see with the recovery, they tend to be a species like canids that responds, they can respond well to persecution in many ways. But so we don't see any evidence of active inbreeding avoidance. Probably it was a small founder effect, and then they just sort of skyrocketed to their current numbers. And both those islands are around 1,000, we think. So.
So thank you for this. So thank you for this talk. I think it doesn't work, but I will be loud enough. So this is very intriguing. The results you showed that with when you have gene flow, then it can happen that inbreeding or raw increases A/B genetic load to counter what you would hope and predict. So I assume this may be because one or a few individuals were extremely successful after introduction and fathered or mothered a lot of offspring that then were more inbred on average when they reproduced. Do you see that one can link this with the number of individuals that were introduced?
Yeah, so that's a great question. Presumably, yes. if you were to simulate that, we could recreate the introduction using mainland runs of homozygosity as sort of the source population and see what would happen over time with some estimate of recombination and mating system. We just haven't done that, but that's a great idea. And so if we can find the time, that would be something we would explore. I think what happened, like you see this with founding events, it's just by chance a high percentage of deleterious alleles were in those founding individuals and became fixed. Not that there was a high percentage. Deer have a high genetic load, and they're just by chance when we moved them to the island, when you're dealing with a dozen individuals, right, drift is the predominant force, a bunch of those deleterious alleles fixated.
Okay, thank you very much for your talk. So I have two very connected questions, so I hope it's okay that I ask both. So the first one is, which method do you use or would prefer to use to estimate genetic load? And then a very leading question, if I were to estimate genetic load based on annotated sequence information alone, what would be your suggestions to incorporate in that setting?
So this is, and I think this is classic, this is all SNP-F. It's a nice program because it's easy to run. My always question mark is it's based off of our annotation and then alternate alleles. So you have to have a good annotation, and then it assumes any alternate allele, like we were, synonymous mutation. It's making some assumptions there that it's bad, but we don't know that for sure. So I think some of these other methods, but the advantage there is you just need your species and your sequencing data. I think some of these that people know way more than me about this. I think if you can use an approach more based on ancestral and derived alleles and some outgroups, computationally going to be much more work, but I think that that lends itself to more reliable estimates.
Yeah.
And then your second question was?
So that's actually why I'm asking, because that's exactly what I'm doing in my project.
Okay.
So keep
going.
So the leading question was basically, since I'm going to use annotations within the model that predicts between derived and simulated variants, then if you have, for example, some like intolerance annotations or evolutionary constraint annotations that you prefer or that you have seen works better than others in combination, or just like if some are superfluous, so to say?
Yeah, I mean, I think it depends on how much time you have, but I think it gets a little bit to Krisha's point with the NE estimates, like run a bunch and, make sure they make sense. So here, that's the wrong slide. I had a rock star undergrad worked for 3 years on this, but we ran GERP, we ran SIFT, we ran SNP-F, and we just wanted to be comfortable with this pattern. But that was, 3 years of his life, and if you're in the last year of your PhD, maybe not a thing.
Time for one more quick question.
I don't know if it's quick, but can you tell us about the methylation response?
Yeah, so I think it's these two slides with small populations, what we're seeing and what we're interested in. And so methylation's all the rage. It's been like that for a while, but now tools are accessible. Inbred zebras, so if you're inbred, you tend to be aging faster. And shrews, so this is a work from my lab, island populations of shrews also appear to be aging faster as well. And so inbreeding, you can you can make the biological connection there. The island, we're not sure, and so we speculate on why that could be. But I think when we're thinking about small populations, and the other cool thing about the methylation data, it's all, for me, compared to genomic data, it's very clean, and the patterns, I'm doing less storytelling, and the patterns are very obvious. And so in shrews, the island, they have a very divergent phenotype, different diet, different phenotype, and when you do your GO analysis, it's coming out as digestive enzymes and skeletal morphology. And But I think this is something to keep an eye on, especially when we're thinking about island metrics that are useful for assessing the health of a population. So if inbreeding is going to increase the aging, or you're just estimating the age structure of a population, not ignoring elevated rates, but if you're a manager, you want to know the sex composition and the age structure. We can't really do that in most animals. But maybe now with non-invasive samples, we can say, okay, 60-40 male-female, and in this isolated population, we're seeing very few adults, something like that. So I think it's a tool that can really help us with small populations.
Let's give Anne a round of applause.